Welcome to this video on Direct Microscopic Examination in Clinical Microbiology. We will cover mastering the gram stain, recognizing bacterial morphology, and understanding acid-fast and fluorescent techniques, simplifying lab science. Microscopy is a critical routine step in microbiology, especially for evaluating normally sterile sites. It guides preliminary species identification, providing immediate data that shapes initial patient therapy before cultures fully grow. The gram stain utilizes a four-step process. First, crystal violet is the primary stain, followed by iodine as the mordant. Next, the decolorizing step uses alcohol/acetone, and finally, red safranin is used as the counterstain. Gram-positive bacteria have a thick peptidoglycan wall and retain the purple crystal violet. Gram-negative bacteria have a thin peptidoglycan layer, lose the crystal violet, and stain pink with safranin. Bacterial morphology guides species ID. Basic shapes are cocci, spheres, and bacilli, rods. Arrangements include clusters, chains, or pairs. Remember to evaluate morphology in the best stained area, usually a monolayer of cells. Gram-positive cocci show distinct patterns. Staphylococci appear in grape-like clusters, while streptococci form pairs, short chains, or long chains. Streptococcus pneumoniae is recognized by its lancelet-shaped diplococci. Gram-negative rods also have various forms. Enterics and Pseudomonas are straight bacilli. Vibrio species are curved or comma-shaped. Campylobacter species are thin, S-shaped or corkscrew. Yersinia pestis shows bipolar staining, resembling a closed safety pin. Gram-positive rod morphology includes large, spore-forming organisms like Bacillus and Clostridium, small to medium forms like Listeria, and short or irregular pleomorphic forms like Corynebacterium. Some, like Nocardia, are branching and filamentous. Acid-fast staining is required for mycobacteria, which are gram-invisible. Their cell walls contain waxy, hydrophobic mycolic acids that repel gram-stained dyes, causing the organisms to resist decolorization and be, quote, acid-fast. The Zeil-Nielsen or hot technique uses heat to help the red carbolfuchsin dye penetrate the waxy cell wall. A strong hydrochloric acid decolorizer is used, and methylene blue serves as the counterstain. The Kinyoun or cold technique uses a higher phenol concentration without heat. The Fight modification uses a much weaker decolorizing reagent, essential for detecting weakly acid-fast organisms like Nocardia, Cryptosporidium, and Cyclospora. The fluorescent acid-fast stain uses auramine and rhodamine dyes that bind to mycolic acids. This technique fluoresces brightly under UV light, offering a highly sensitive method for rapidly screening acid-fast bacilli. The acridine orange stain is a specialized fluorescent technique that detects both bacteria and yeasts. It is ideal for smears from blood and body fluids, especially when organisms are present in very low concentrations. A critical interpretation error is inadequate decolorization during gram staining. This causes host cells and gram-negative bacteria to falsely retain the primary dye, appearing blue or purple, leading to incorrect preliminary diagnoses and improper antibiotic therapy. In summary, specimen type dictates the staining technique. Routine specimens use gram stain, suspected mycobacteria require acid-fast, low-concentration fluids use acridine orange, and rapid screening is done with auramine rhodamine.